Sir, shall we start? It's 4 now.
Yes, let's start.
Okay, sir. Thank you, sir. Good evening to all, and welcome back to the second day of 3 days online workshop on Arbuscular mycorrhizal fungi, taxonomy and biodiversity, taxonomy and propagation. And to move forward, may I call upon Professor R.K. Singh, Head Department of Botany and Chairman Organizing Committee of this workshop, to formally welcome all the participants and the speaker. The speaker is here, sir, ma'am. Sir, over to you, sir.
Very good evening to all of you. As Evelyn has said, today we have the second lecture by Professor Manju Gupta. Yesterday it was the lecture delivered by Dr. Mahavir Sharma from IARI, and the institute was Indian Soybean Research Institute, Indore. So I welcome Professor Manju Gupta. Perhaps he is abroad now and she is speaking today from there itself. So welcome, madam, to this workshop.
Thank you. Thank you. My pleasure.
We are very thankful to you for accepting our request for delivering a lecture. Before introducing you as a speaker today, let me first introduce my department to you. We have 9 faculty members in the Department of Botany of Rajiv Gandhi Central University at Itanagar. And out of 9, 3 have been awarded PhD on Am Fungi. And under them, at present, 4 research scholars are working on Am Fungi, looking at different aspects of Am, particularly the biodiversity. Out of the 3 faculty members who did their PhD on Am, one is myself, second is Dr. Evelyn, and third is Dr. Oidai Nimaso. We have a very good lab for research work on Am Fungi. The other faculty members, 3 more faculty members are on microbial ecology and microbiology, and remaining faculty members, they work on diversity of plants and pharmacognosy. So with this brief introduction about my department, I am now introducing the speaker to the audience. Professor Manju M. Gupta, presently serving as a professor since 2018 in Aurobindo College, University of Delhi, has joined the college in 1992 after completing her PhD in botany from University of Delhi in 1992 under the very renowned academician Professor K.G. Mukherjee. Her PhD was in the field of microbial ecology. During PhD, she was awarded GRF and SRF of CSIR. And just after completing her PhD, she got Vice Cost Fellowship in 1996-97 from DST and did her postdoctoral research in Department of Plant Science and Soil Nutrition, University of Western Australia, under a very reputed mycorrhizologist, Professor L.K. Abbott. Thereafter, she did another postdoctoral research at JNU under Dr. Devpriya Chaudhary in 2001-2003. She was appointed in college 1992, and thereafter she developed a very good research lab in her college, where almost all facilities for research, PhD research on mycorrhiza is available. She has completed 3 research projects and has developed one online biodiversity database on Am Fungi present in India. She has been head of the department for several years in the college. She has visited several countries, particularly Oregon State University, USA 2018, where she chaired a session at International Symbiosis Society Congress in 2018. She has visited Czech Republic, Prague in 2017. She attended International Conference on Mycorrhiza, Conference number 8 in Northern Arizona University, USA 2015. She delivered a keynote address in 8th International Symbiosis Society at Lisbon, Portugal in 2015. And she also attended 11th International Fungal Biodiversity Conference in Germany in 2013. One more conference that she attended when she was research scholar in 1990 at Vienna, International Conference at Vienna. She is member of several societies. The most prominent one are International Symbiosis Society, where she worked as vice president, International Society of Mycorrhiza, and International Society of Computational Biology. She has several publications to her credit, almost 30 publications, and 5 books, edited books. She has been member of editorial board, particularly of Journal of Symbiosis, published by Springer since 2019, and Journal of Recent Advances in Applied Sciences. Yeah, Applied Science. So she has a vast experience, research experience, particularly in the field of mycorrhiza. So I am glad today we'll be listening to Dr. Manju Gupta on taxonomy of Am Fungi. Thank you. Now
Thank you.
I invite Professor Gupta to kindly deliver your lecture.
Thank you, Dr. Rajiv. I am humbled by your kind introduction. I think there was no need for such an exhaustive, but you covered all the things. And I am delighted to know that faculty in your department, they are working on mycorrhiza. And I was, I really liked it. Meaning 3 of your faculty members work on arbuscular mycorrhiza. And I feel it really nice, because I was just wondering how much of the trained mycorrhiza people are there who will be able to appreciate like whatever we've been talking, because at many places it will become specialized. So I thank you again, and I thank Dr. Evelyn also for inviting me here to give this talk on arbuscular mycorrhizal fungi, and especially on their identification, because the topic is very close to my heart. And meaning at several places you would feel that it is getting spontaneous, and thanks for highlighting, but I've been given a good exposure from maybe the God side or just by the availability, that for last, at least if I'm not traveled at many places in India, but abroad I have traveled in almost very good laboratories, and I have interacted with maybe Joseph Morton, Chris Walker. I usually keep talking, like in my talk, you would know that last week he has sent me a paper and inoculum of Am Fungi, which is being released in India. He said, what do you think about this? So I thought, because this topic, though this time was a bit, it's 5:35 in the morning at New York. I was on my family holiday, but still I thought I'll be able to manage and give this talk, and I hope that you'll find this worthwhile. So do I begin now? I start with my slideshow straight, right? Thank you. So, am I audible to everyone? And you can see my slides. Is that
yes, I'm
right?
audible, and
the slides are seen by everyone. Right. So, as Dr. Rajiv told, we'll be talking about methods in identification of Am fungi. And I'm glad to have such a audience where we have most of the research scholars and PhD students. But the thing is, another good thing here in the audience is that a lot of people are trained faculty in mycorrhizal. So I planned this talk from here itself, from New York, because I just missed that I have to give this talk. So I didn't bring many of my tools from India here. I left India in December. So, but still I have tried to organize this. But while planning this talk, my objectives for this, and especially I was told that this is a workshop and we have a diversity of audience, meaning they may not be from mycorrhiza also. So I kept it, meaning to a bit, for beginners maybe, or I wanted to generate the interest of the budding scientists from different fields to these tiny friends of ours, which we call as arbuscular mycorrhizal fungi. We'll be talking about them in details, and I want to make you familiar with at least with the structures of few common ones which we find here in India, so that when you leave this workshop and when you go from here, at least you are conversant with few of them. I want to familiarize everyone with the standard protocol which we follow for identification of Am fungi. And I'll just take you to several useful resources which are very useful in identifying this fungi. And I'll tell you about our work, what we have done for a few years, like when we've been given a few projects from UGC and few international projects where I'm a part. So let's begin with this. And as usual, most of the times I start my talk with a joke. And again, a joke, because I'm in New York, so I like to start with a joke about New Yorkers, on India too. Basically, we hail from, and we think about India all the time. So again, I couldn't find the mycorrhiza people, and I didn't want to introduce them in the joke. But the joke talks about these archaeologists. The archaeologists from France, and they dug down 10 feet down, and they found what? They found that there were copper wires dating back 200 years. They concluded that the French ancestors of they were having telephone network which was 150 years old, meaning the French people were using telephone network 150 years before. So not to be outdone, New Yorkers felt a competition, and they followed. They dug down 20 feet instead of 10 feet, and they found that they had copper wires which were 250 years old. And then in the New York Times here, there was a news saying that the high communication network has been discovered, and it was discovered 50 years earlier than it was discovered by the French people. It is usual in science that we have competition, we have extrapolation of things. And when this news reached to India, what did we do? The archaeologists from India, next week in the local Delhi Times, they reported that after going down to a depth of 33 feet in Delhi, they found absolutely absolutely F the prom, and therefore concluded that Indians 250 years ago had gone already wireless, meaning there were no wires reported, and they could interpret that we were wireless. Yeah, I think it was a laughing this thing for me, and because I'm going to tell you similar situations, good start from my side, and I'm going to tell you several similar situations that how the studies in mycorrhiza nowadays, they are better, but just few years before how they were the international joke. So let's begin with this. And I start with straight about my work, which I started in 2013. And in 2013, we took up a project. It was a small project, Delhi University funded project, with undergraduates, and we began with analyzing the state of Am fungal diversity in India, and we studied this at several states. Sorry, I couldn't hear. Any problems with this? I hope there are no problems and no questions. So we were doing it in India, and we collected samples from 50.
Ma'am, you have muted yourself. You have muted yourself. We can't hear you.
So since the beginning it is muted, or this has just happened?
Just happened just
Okay.
now, and the slide has gone, ma'am.
Slide is gone now. So you can see the slide now?
Yeah.
And this is where it went muted, okay?
Yeah.
Right? So I start from here again. So I was talking about my work and where we were analyzing and assessing the status of Am fungi and fungal diversity in India. As Professor R.K. Singh told us, I have two backgrounds, One is the Am fungi, of course, that lies very at bottom of my heart. And the other one is bioinformatics, meaning I have trained myself very well in computational resources apart from these fungi. And these studies, this sort of knowledge and background really helped me. While studying these, assessing this Am fungi, we were using all computational knowledge which I had by that time. And we collected all these 64 references, and we had our own samples also. And what we concluded is that we have 148 species of Am fungi occurring around in India, which have been reported properly, and they belong to 21 genera, which is much less than 341 described from the world in total. So we just had 148 at the time, and we found that 7 of these belong to Funneliformes, 1 to Septoglomus, 48 to Glomus, and maybe 27 in Acolospora. So from that paper of ours, maybe it can be seen that they belong to 21 genera. And we also, we could do the quantitative analysis. We could calculate the abundance of different species apart from their geographical location. And we found that the Glomus is the most commonly occurring or most commonly reported species, the genera, and it is represented by 48 species, which was followed by Acolospora and Scutulospora. We had Funneliformes mossae and Rhizophagus fasciculatus. Those were reported to be the most common species. And I told you we did it with MLXS and SQL Server, and we could analyze the abundance at every step. But I tell you again, where is the joke and what was the problem and what was the situation that was existing? It was, embarrassing as well, apart from, I could say, joke, that it was not easy to conclude that one species like Funneliformes mossae and Rhizophagus fasciculatus are the most common species in our land. How did we do that? So the first confusion or the first problem which we faced at the time, that like for Funneliformes mossae, I would say, in literature, people by then, though it was changed, its name was changed to Funneliformes mossae, but people would report it by Endogone mossae or maybe Glomus mossae. Same was with Rhizophagus fasciculatus. Though the name has been changed based on the phylogenetic classification, but people will still find them and they will report them Glomus fasciculatus. Their previous names like Glomus fasciculatus or maybe Rhizophagus fasciculatum and or Endogone fasciculata, but the name which was properly given to this was Rhizophagus fasciculatus. So The problem which we reported was that we have to have one name for one fungus, first of all, popular among the scientists around. And the second thing is the name should be correct. See, that was the problem about the nomenclature. That was not the only problem. The other problem which we found that the names are changing so rapidly. Meaning, I said we have to have one name for one fungus, and we reported that most of the glomus genera have been renamed. Like Glomus mossae, we used to call that as Glomus mossae, but now it is Funneliformis mossae. And we have to call them by the name which is accepted by the scientific community around. Then I tell you one more funny thing is about these changes in the names were so rapid at the time, like the Glomus intraradicis, which we call as model organism when we go for genome sequences of arbuscular mycorrhizal fungi. That's the model organism. And when this project started, it was called as Glomus intraradicis. But before the project could be completed, it became Rhizophagus irregularis. And before that, the name was changed to Glomus intraradicis and then Rhizophagus irregularis. And in between two names, so two more names came. One was Rhizoglomus instead of Rhizophagus. But now finally it is said to be called as Rhizophagus intraradicis, even not Rhizophagus irregularis. So there was so frequent change in the name, and that was coming. Why? Because the authority, no proper, things were getting followed. So the other thing which we reported is that the names are changing so rapidly, we have to have one name for one fungus. I have taken just few snapshots from few papers, which I thought would be worthwhile when we're talking of these fungi to be mentioned. Like there's one from this paper, Redeker et al. He reported this statement, he said, people are studying the identification of fungi. They're talking about Am fungi. They are giving them names. But when the names are being explored, like in a recent case in which the type is almost uninterpretable, is that of Otospora boreae, which was described by Ohel et al. And it was examined on loan from the herbarium in Zurich. Ohel, everybody knows that he works in Zurich laboratory, and from where this spore was lodged. And when they were asked to provide the specimen so that it can be explored further, the specimen available were dead. They were in very poor condition, and they could not be explored further. So when this new name was coming, it was uninterpretable for the other scientists. Another example reported in the paper, which says a recent species of Denti scutata, nigerata, which came from India. And they said the holotype, it is written that it is holotype and it is deposited, but on personal inquiry from S.W. Khat and P.K. Sharma, they told that no such specimen exists in the Goa University. I just don't want to offend anyone, and with due respect to all the scientists occurring around, I am just trying to explain the situation which existed in studies of mycorrhiza. I'm not talking of very old situation. I'm talking of 2013 and 14. Right? This was another thing was being reported. And then I met in 2017, there was a live symposia session about this phylum Glomeromycota. Should we call this as Glomeromycota, or this is subphylum Glomeromycotina of phylum Mucoromycota. See, I just took this picture of mine. I was not in the position to talk much about it because they were very senior faculty, but I still thought I could introduce you with, I have a cursor here. This is Ruther Schussler. He was there in this symposium because he described this phylum Glomeromycota. He raised this phylum Glomeromycota in fungi. This is Redeker, Dirk Redeker from France. He is basically, presently, is a very renowned taxonomist apart from Chris Walker and Joseph Morton. And he was also there, and both of them believed that we should keep this Glomeromycota and we should not shift this to the subphylum Glomeromycotina. This is Mark Brunred. I worked with him, University of Western Australia. Apart from Lynette Abbott, he was also there, and Dr. Chris Walker was also there when I was in UWA. This is L. Ledreso. And in this, this symposia, I tell you, one more scientist was there. She was Marza Opek. We all know about Marza Opek. She from in Estonia, University of Estonia. She works on environmental samples. She works on the virtual species or the OTUs of Am fungi. She works on environmental samples, and like the described species are 341 even now, but she would say the mycorrhizal species, Am fungal species belonging to this Glomeromycota or Glomeromycotina are above 500. And how she says this is because the sequences, on the basis of sequences or OTUs, she says this represents to a species. But unless the species is present in its morphology, we cannot identify. So that was another vicious circle which is going on, is that these environmental people are going on describing species. This morphology people were going on describing species, and there were different primers on the basis of which the classifications were coming. There were 3-4 classification. But this symposia, for me, it was a matter of pride to become a participant in this symposia in ICOM 9 2017. And I really enjoyed this in a way that I could see that how the top-notch scientists, they discuss and they have problems, and how they raise the phylums and then they delete the phylum. Though in this symposia, it was somehow, decided that we could abolish use of Glomeromycota, but what happened after a while? I see these changes, the scientists didn't agree. In 2019, again, I read that the scientists say we do not adopt to exit this phylum, to abolish this phylum Glomeromycota and use the word Glomeromycotina instead of Glomeromycota. People still prefer to call this as Glomeromycota on the basis of phylogeny, and they find this to be consistent. And even the school of thought to which I belong, I also believe in phylum Glomeromycota, where it falls in as the level of phylum. So from now onwards, everywhere you will see this written as phylum Glomeromycota. So that was another point of confusion or the dicey situation existing with the things in mycorrhiza. I come back to another situation which I reported existing, is again a highlight from a paper, is that people pointed out, even in the conference, people pointed out that the species description or the publications are not being properly reviewed, or they are in haste. So the statement exactly which they said is that in this context, we would like to address some comments to the editors of the journals who are publishing papers on mycorrhiza, to send to those referees who are doing their duty of reviewing critically, and especially when the new taxas are being proposed, because the phylum of Glomeromycota contain many species that are of uncertain phylogeny, because to isolate DNA from them was a challenge, and unless the species are described on the basis of molecular characters, they cannot be accepted or identified. So that was another thing which was being reported, that the species description or publications, they are coming up in haste and they are not getting reviewed critically. So there was a request to the referees and publishers that it should be critically reviewed and the type specimens is to be checked thoroughly. Then another paper in 2018, this is 2018, by again the Mark Brunred and Leho Tedrose, whose picture I showed you just now. They said is that misdiagnosis of mycorrhiza as an improper cycling of data is leading to false conclusions, and he also cited several examples. So this is the scenario even now. But now I feel that it's better once we have understood, at least for us, we have decided one school of thought that we will call this as phylum Glomeromycota. And I have a list of species released by Chris Walker, my supervisor in my postdoctoral research at University of Western Australia, and that we have one name which is accepted name for all these species and species list which he has prepared, and we follow that name while naming any AM fungi, identifying any AM fungi. So let's, with this scenario, I was just trying to explain you this scenario, what is existing, what was existing around us, and what is existing even now. And it was for us to decide what we want to do and how we want to identify, what name we want to give to the fungi which we are collecting and isolating and trying to identify. So let's begin with this. I was just explaining this scenario, what is happening around. So a lot is happening with AM fungi, and all the AM fungi belong to phylum Glomeromycota from my side, but people are still shifting this to subphylum, meaning you will see the reports in 2019, 2020, where people are reporting it to be a subphylum of Glomeromycota in the phylum Mucormycota. See, there was another group of AM fungi, maybe I'll talk about them later. They are also shifted from Glomeromycota now, but they've been done on the basis of proper phylogenetic studies. And then they were, we have few classifications existing. One is by Joseph Morton, one is by Schubler and Walker, I told you, in 2010. And one is based on LSU rDNA sequences, one is based on SSU rDNA sequence, and there's one more by O'Hill. I told you the Zurich, that's another school of thought, and his classification is based on morphology and phylogeny both. But to me, and generally, I found the classification, and we somehow decided to follow this classification given by Schubler and Walker 2001, which is based on SSU rDNA. And I'm going to tell you the methods, how we identify. It's a workshop. I have kept my focus on methods, and I'm just trying to build a background for explaining my methods. You know, what is happening is that I believe there are few participants who are not necessarily trained in mycorrhiza and may not be from fungi. So I try to keep that simple so that everybody appreciates and is ready with questions, whatever they want to ask. So AM fungi, based on their presence of arbuscules, earlier they were called as AM fungi, but because our vesicle is not always present, V is removed, and now we call this as arbuscular mycorrhizal fungi, because arbuscular is the feature which is present in all the AM fungi. And I told you there exists a lot of turbulence about their names, taxonomy, and publication of species and the type specimens which need to be deposited. So again, we came to the consensus classification where there was a paper by Dirk Redeker, Luther Schubler, Hert Stroginger, Sidney L. Strumer, Joseph Morton, and Christopher Walker. I really like this picture of mine with Joseph Morton, whom I met at 8th ICOM in North Arizona University 2015. And these ICOM people had invited me for my talk there. And it was just a pleasure and a blessings from God which came to me. And again, I organized a talk by Professor Chris Walker at my institute. Again, because of this online and COVID times, I could do that on 4th May 2020. And Professor Chris Walker, they joined and they addressed our students about the problems and this nomenclature self-code, meaning that how important the type specimens are and how should we maintain them and which code is to be followed. So with this all background happening around me, we had the consensus classification which need to be followed. So I'm now going to tell you what are the main features of this consensus classification broadly. Is that it believes in monophyletic phylum, Glomeromycota. It's a phylogenetic classification, and the phylogeny could be traced back. And we have 4 orders in this: glomerales, paraglomerales, archosporales, and diversisporales. We have now 341 species, but when this consensus classification was proposed, by then it was having 228, and now those at that time were grouped into 25 genera, but now we have more. But glomus is not a monophyletic clade, it's a polyphyletic in itself, and lots of genera from glomus are getting changed, meaning they are being redescribed and given different names. They are still in the process. I can tell you several examples where the glomus is changed to Funneliformis, Septoglomus, and glomus. There are several names coming up from this based on phylogeny. And the conclusion of this consensus classification is that the morphospecies concept is difficult in AMF, and it must be combined with the phylogenetic concepts. So we must have sequences before we describe the species. This is what was made mandatory at the time, and after that, meaning it became a bit more tedious working with Am fungi, in a way that earlier also there were lots of problems, like the structures of Am fungi, they're not consistent. The spore walls you cannot observe very often on the basis of which we identify. But to get DNA from them, to amplify and then get their sequence, again, another challenge. So how we address that challenge and how do we go about it, I'm just going to tell you that in details in the second part of my talk. But in this first introductory part, what I'm trying to do is I'm just introducing you these Am fungi. I will try to tell you about few common genera, what do we observe in those, and then I tell you what is the scenario existing. Again, with the help of some of my publications, I'll take this forward and just tell you where do we exist now, and then you'll be able to appreciate the type of work which we must undertake when we're talking of Am fungi. So again, this picture, why I have taken this picture and why I want to show you here is that we all know that Am fungi are ecologically and economically very, very important. And yesterday itself, I was attending Professor Sharma's lecture. I just came in, I just wanted to listen to what is happening. And it comes in the market. It is available for all the farmers. And this is one example of a inoculum which is being sold in the market. And what does this write? I took this print screen, this snapshot from the market, the website of these people. And why I especially chose this, because my teacher, Professor Chris Walker, he sent it from there, from Royal Botanical Garden, Kew. He said, can you look into this, that they say they have 3% Am fungi, and they have in this present this glomus micro aggregate tune, right? So do you see this name anywhere in the list, or is this species being described by anyone before this one? So this is the state of market of Am fungal biofertilizers which are being sold. And there are a few papers taking rounds. I'm just talking about taxonomy here, but if you talk about their economic importance or their use as biofertilizers, we have to be very cautious, not only about their identification, about their productivity. So there was one paper of this mine which came in 2020. I critically reviewed this scenario of economic assessment, meaning economic application of Am fungi, and it was a privilege again to work with Lynn for this paper. And we critically analyzed how would we can explore the economic benefits of Am fungi, Am fungal symbiosis. This paper was published in Symbiosis, where I happen to be in the editorial board. So it was an invited paper, by the way. So what happened is, what did we find in this? What did we report? Is that, see, not every Am fungi is giving you positive result. The benefit of Am fungi are context dependent, meaning in certain situations they will be good, they will give you growth yield, but there are equal situations where it is going to give you growth depression. So it is context dependent and could also lead to growth depression. What we must consider before applying these fungi in the fields is the native microflora, meaning from ecology point of view also, when we apply any inoculum in any agricultural soil, we are disturbing the ecology of that agro-ecosystem by introducing an additional fungi. We must consider the native flora for these inocula production and how we must understand what flora is present and how do we multiply that. So again, we come to the same question, the taxonomy. We will not be able to go far without understanding the taxonomy of these fungi. So this picture on the left, I just selected because I wanted to tell you there is a range of benefits which have been reported from these fungi, but I'm coming to the latest that it is context dependent and it could lead to growth depression also if it is not, if the native flora is not studied and it has not been checked properly before being applied in the fields. So I come back to the business now. Well, we'll be talking about the taxonomy, and in this introductory section, that is the first half of the lecture, where I'm trying to tell you is the Am fungi taxonomy. I could make the background that it could be very confusing. You have to decide for yourself how you are going to do it, and to get benefit of to get the maximum benefit from these promising fungi, we have to be very careful and in touch with the people who are expert in this. So I begin with the structure now, taxonomy description, and here, I'm not coming to the actual taxonomy, how we are going to do this here. It's a workshop, and I planned it in this way that I'll tell you all the steps and how do we do it, especially at University of Delhi. So, I'm just telling you the introductory part so that those who are not from Am fungi know what is happening. So this is again a drawing which I picked up from my paper, which I wrote myself. I was the sole author. I've drawn this picture and it tells you the complete structure of Am fungi. The spores can be, so can you see my cursor if I am, yeah, I think my cursor can be seen by everyone, is it?
Yes, it's visible,
Yeah.
ma'am.
So these spores can be, because I'll be using these terms now very extensively, and again, those who are not from the field would find it difficult to appreciate. So I'm just quickly picking up the terms, and so that it just goes fine with everyone. So these spores, they are usually present in the soil, they germinate, and they produce intraradical hypha. The hypha inside the root is intraradical hypha, outside is the extraradical hypha. We have vesicles produced inside the root, and the vesicles could be produced outside also. This is the spore which is produced inside the root, meaning the spore can be intraradical or in the soil both. We have vesicles present and we have the eponymous structure, that is arbuscule, and we have spores outside. They can be many, they can be one. There's a subtending hypha, the hypha attached to the spores because the spores are dispersed with the hypha. And it has a role in soil aggregation. If you want to study its functionality or glomalin, you have to have the soil around, that is the aggregate, the glomalin cluster. And it is on the right side, it's a small cleared preparation stained with trypan blue. And so what is happening is, just give me 1 minute because my laptop is going to go. I am at the new setup here, and Yeah, I think, yes, just hang on 1 minute. The battery is about to go. So it's back now. Yeah, so just sorry for the interruption. Now it's better. So I have to shift a bit. Yeah, so goes fine. So that was it. So we could see all the structures very easily in the laboratory and appreciate all the structures. Again, the same picture, extra intraradical hypha, vesicle spores, extraradical hypha, germination, and everything. So that was the structure generally, and I just wanted to pronounce the terms which I'll be using throughout the workshop. Then just wanted to make you familiar with some of the genera and how they look like. Is the most popular one, most common one is Glomus. Then we have Archaeospora, Redacra, Pachyspora, Acolospora, Rhizophagus, Claridioglomus, or Rhesocetra. I thank Professor Walker for giving me these pictures. And we have we have Paraglomus, Diversispora, Sclerocystis, Scutellospora, Ambispora, Funneliformis, Gycospora, and Trophospora. So they are the common genera which you will encounter throughout the talk. And but we'll be mainly talking about glomus. And I like to explain the structure, what to expect when I'm talking about the glomus throughout the workshop and when I'm telling you the procedure or the methods of identification. So the glomus, glomus would have a subtending hypha, like you see it here. It's a drawing. You see it here, but it is not swollen, because in contrast, in the other fungi, you would see it swollen. So you always have subtending hypha present in glomus, but it is not swollen here. And the other thing is that in glomus, you can see the spores inside the root also. And the other thing here is that it can be single or it can be in clusters. And you have one or more wall layers. So what do we observe when we talk about the taxonomy? The subtending hypha, spore, its wall layers, and the content. So it's a small cluster of Glomus macrocarpum, which was isolated by one of my undergraduate students. Then I will also at several places talk about Gygaspora and Scutellospora. Now, what is special about Gygaspora? Meaning they are large spores. Their size is usually greater than 200 micrometer, and it goes up to 1000 micrometer. They have bulbous, I was telling you just now, just by the size, because they're huge in size, and they have a bulbous subtending hypha here. That is the main feature on the basis of which we could identify. So whenever you see the pictures again, you should observe for this bulbous hypha. And it has a germination shield here when the spore is germinating, because for identification, we use germination of spore also as a character. And when you observe germinating Scutellospora spore, you will see a germination shield like this before germination. And you have auxiliary cells also present, and they are present in the soil hypha. I can show you the structure of that also. So these are the features of Gygaspora and Scutellospora. Both of them fall in the name of the giant spores because they are very big in size, meaning up to 1000 micrometer. And they have typical structures like germination seed and auxiliary cell. So just to revise the terms in taxonomy, so I think it made sense. And now you know what is the auxiliary cell. It is produced in the soil hypha, and we have a germination shield in the germinating hypha of Scutellospora and not Acolospora. So this again a picture of Gygaspora, and you see the swollen subtending hypha, and you have auxiliary cell present in the soil hypha. Germination shield is absent here. In Scutellospora, you see several ornamentations very often, but it will show you the auxiliary. So for identification of Scutellospora or for Gygaspora, we need to perform the germination test. And otherwise, just based on the morphology, you find them very often, because they are very large in size, you can identify them quickly. At least to the general level, identification is not that problematic, and you can identify them. So other two common genera which I chose to tell you or show you is Acolospora or Entrophospora, because I cannot take all of them. But still, when you are attending a workshop on taxonomy, you must be familiar with few genera at least. So what is happening is that in Acolospora and Anthropospora, both of them are sessile. So when you see them in a soil, you don't see a subtending hypha with both of them. And they are formed by a different method, meaning they get their nourishment from this sporiferous saccule, which is usually reflected in their wall layers. But their method of meaning attachment or detachment from this sporiferous saccule is different. On the basis of this, they are kept in different genera, and also on the basis of phylogeny. So if we say Acolospora, Anthropospora, both these spores are sessile. You don't see any subtending hypha in both of these. And the saccule loses its saccule is lost after, when the spores are mature. You don't see any saccule in Acolospora, but you could see in some of them in Anthropospora in the pre-differentiated shell. So by telling you all this, what I could tell you is that you will mostly see glomus. You will be able to at least pinpoint in your light microscope that this could be gloma, this could be Scutelospora, or this may be Gygaspora, just basis of the size. And if you don't see any subtending hypha, you could say maybe it is Acolospora or Anthropospora when we have no subtending hypha. And then we go into the details of making slides, and that I'll tell you in exact steps. But still, I could form a image in all the participants, in all mind of everyone, what to look for when we are talking, at least the common genera, which we have distinguishing feature. There's another thing, where I worked at the University of Western Australia, which is included in mycorrhiza even now, arbuscular mycorrhiza. Those are fine endophytes. Earlier they were kept, this is my paper with Lim again. I always take this privilege in publishing papers with Lim. And is fine endophytes. They earlier were kept with glomus tenuis, but now they've been removed from there. And their feature is that their hypha is very thin, roughly 1.5 micron in diameter. These are a few pictures from my paper because it's an old paper and we published it in black and white. So the branch produces, meaning it has a typical fan-like structure. But the most important thing is that, like Scutelospora is the other range of the giant Am fungal spores. This is the other side where we have less than 20 micron of spore size. But now they are removed from Glomeromycota based on phylogeny. They come under Zygomycota now. So with this one, I just like to wrap up this introductory session. And I think how much time I have taken for this, I would just like to know. Where's my phone? So I'm just on time. I took half of the time for this one, and I'll devote rest of the time for the methods or the procedure. For procedure which we follow for identification, or maybe the exact steps how we do it in our laboratory. I'm going to demonstrate you that online. But before I go further, I hope I have made a background of the scenario, which classification to follow and why follow, and what to look forward to inside the root or the soil and what to collect. So the forward with this, ending up this first section of my talk, is that I'm saying that the time has come and the taxonomy is not easy, especially the taxonomy of Am fungi. We have to follow the morphophylogenetic classification. And I pointed out that we follow the classification given by Ruther and Shubler and Walker. And we believe in the phylum Glomeromycota rather than keeping it to subphylum Mucoromycotina, but it becomes your personal choice, because at the level of phylum, it doesn't disturb anyone. You can decide accordingly which one you want to follow when there are so many people proposing lots of things. And I also, I can show you while identification that we follow the names given by Shubler and Walker in the species list published in 2010. So with this, we reach to the next step. I could attempt the questions if anybody wants to ask any question. Otherwise, I straight come to the method, how we identify the Am fungi in the morphophylogenetic spore identification which we follow. We go ahead. Yeah. So when there's no response, I hope everybody is listening. I can't see anyone. The audience is here. In this Google Meet, this is the problem that I cannot see the audience. But in Zoom, sometimes you can see. Okay with this?
Kindly proceed.
Yeah, thank you. So let's go forward. So we are now talking about the steps in present step, which must be followed when we are going for the identification of Am fungi. And the first important step here is obtaining the AMF, right? And in obtaining the AMF, we usually start with the field samples, because if I'm starting my study in Delhi, I will go to the fields in the Delhi and I will see which all fungi are existing here. Or maybe sometimes when you are exclusively working on fungi, you might start with a culture with already, you know, the trap culture where the spores have already been multiplied. But whatever you start with, you must have the live living material with you. So if you are starting with field samples or with maybe the cultures, already cultured samples of spores, you must pick the live samples. Like by this stage, you know, I mean, is for, you know, for DNA isolation also, you will not be able to do anything. So the first step in obtaining the good sample for AMF is that you should be able to distinguish between a live spore of AMF and a decaying or the dead spore, because that is going to trouble you and that will lead to inconclusive results. And again, you will not be able to isolate DNA from those. So you have to have clean, live specimens present with you, with whichever fungi you are working. If you're working with the field samples, again, you have to have the live specimen. So we must not use these spores directly from the field. Why? Because they are not healthy, they are not viable. Meaning, it's a very important caution which is coming from my side, because I see a lot of papers, especially when I was studying all the papers which are existing in India. The pictures will tell you clearly that they are dead spores, but still they are giving a name to that. So we must not use these spores which are coming from field directly. At least 2-3 generations, they must be cultured in the laboratory to get good, clean, living spores. They will not be healthy. They are also not viable very often. And they're usually parasitized by other microorganisms. And if you try to observe them, you know, they have lost all their taxonomic characters, like soil chemistry, temperature, etc. They change, the microbes present, they change their appearance. So it will lead to wrong identification. And they also give you wrong data in a way. They just represent those species which are able to colonize at that time. They were able to colonize and they could be reported. But there are several species existing in that situation which would be missed out if we don't make a trap culture. So after having procured this specimen, like if you are taking a culture, purchasing a culture from lots of places which we have now for already identified species, or if you are beginning with a field sample, there is no options. You have to raise cultures. Again, that's a picture for single spore cultures, which was raised in our laboratory. We usually use maize for this one. So because Am fungi are obligate symbionts, we all know. So no exenic cultures are possible. We must go for, that is why, you know, people go for shortcut, because it's a very lengthy and a cumbersome procedure. So we must go for pot culture. And in pot culture, we have a very high, because it's a very labor intensive process and time consuming also, meaning you have to have a sterile soil. And because I remember in UWA, I used to sit in the growth chamber in their poly house, and then I used to buckets of the soil. They used to have a very big autoclave all the soil and then fill them in the pots and then water them so nicely. So raising culture was a very tedious job. And it requires a generation time of 10 to 12 weeks depending upon the host species. And variety of hosts can be used, but I said we usually use more maize here in India, meaning in my lab at least. And we might need to do them for several generations if we are especially starting with the field samples. And we have different options for raising cultures. It's your choice. Maybe you could use simple soil trap culture where you take the soil either from wherever is your interest lying, if it's a culture or if it's a field sample, you take some part of the soil and then you transfer this to the already sterile soil or any other material, any other medium which you are using for multiplying your inoculum, and then you can uh, grow a host plant in there, either seeds or seedlings, whatever you want, and then wait for 1 to 6 months and you get a culture. But I tell you, this will not be if you are using a already cultured, it's okay. But if your sample is coming from field, it will be a mixed species pot culture because the soil will have many spores. And again, some will multiply, some may not multiply, but it will be a mixed species pot culture. And when we add the soil, what happens is it brings in lots of other microorganisms along with this. Like if you are adding, so what we used to do in our laboratory is that we used to, by wet sieving and decanting technique of Am fungal spores isolation, we used to wash them thoroughly. We used to isolate these spores by this method, and then we used to wash them thoroughly, pick those spores which looked good to us, and all these spores in that 50 gram of soil, all these spores, take them in an append off, and we used to raise mixed culture with that. Even then we used to have infection because the spores in the field sample, they have other organisms present. So in the first generation you get a mixed species pot culture, and you have to raise it to several generations if you want to identify, you want to use it for taxonomy purpose. Because I was doing the abundance study also, so for me it made sense like several times. So at least you should have up to 100 spores with you if you want to study the taxonomy of that, and they should be clean and needed to be sorted out in the dissecting microscope. So in the next generation, what you can do is your mixed species culture, you could use as a trap and grow them again, you following the same method. And what will happen is the spores, they will germinate, and some of the spores which were dormant earlier, maybe they germinate this time now. But still you need to do it for 2-3 generations, otherwise you will always have this problem of infection or contamination in your cultures and leading to long conclusions. You don't observe these spore walls very clearly if you have contamination in your cultures. Again, I'm talking about the different options, how you can raise the culture, but we make trap cultures. You can make plant trap cultures where what you do is you just remove the plant. If you want to study the native flora, wash the roots thoroughly and remove all the traces of the soil, external mycelium, and then plant them on a suitable substrate. Meaning already in the pot, you have a sterile soil that is there in the growth chamber. And what you will do is you will come with plant of your interest, dig that plant and wash that thoroughly, plant that in this new pot and wait for the time this may be a little quicker method, but again, you would need to do this for 1 or 2 generations. Similarly, in Australia especially, it is very popular to use root fragments. They use root fragments. If you see them, they are quite easy to use also, in a way that what they do is that, like I have a culture, say of any species, they will dig out their roots, dry them, chop the top portion away, cut them into pieces, and use these root pieces as a source of inoculum. And it has the same advantages of same as trap culture, but it would give you less number of species. So you will quickly reach, to maybe few species culture or mono species culture. So there's another option which I just missed out while writing this is monospecies culture also, where what you do is you raise the culture of one species, meaning in the dissecting microscope, I told you the characters of glomas, at least you can make it out. This can be glomas or this can be scutelospora. They look very large in the dissecting microscope. So what you can do is you hypothetically you can visualize that this could be one species, multiply them, and then by one generation maybe you would know that now that species has grown. So then you can make a culture of one species only that we call as monospecies culture. So there's another option which I have written here is single spore cultures. What are single spore cultures is when we raise a culture for one species from a single spore. We could, because in single spore culture is, I said earlier, we could keep about 100 spores as inocular, but in single spore culture, we use only one spore as a source of inoculum. And what happens is that spore may be dead very often. So you have a very poor success rate in this, about 2%. Or that is why we had to raise so many plants, so many pots, and we also got, but we could raise for a few species these single spore cultures. And they are the main ones on the basis of which we could identify the taxonomy. For taxonomy especially, I would say if we could raise a single spore culture, but the limitation is that not all spores are going to multiply and give you a single spore culture. So this is the best, otherwise you have to manage with monospecies culture. At least you should have that if you are talking about a species identification. All right with this. So I had written few tips that if you are going to grow cultures, if you're planning to raise cultures, you must, follow the several ethics or several shortcuts. And because it's a lengthy process, if something goes wrong, your 3 months and 6 months are gone without anything. So we must wash the propagules very thoroughly before we use them for a starter culture. And the normal source of contamination for in any fungi lab, all those precautions need to be taken, meaning it should not happen that you are touching the pots or anything with finger to assess the moisture level or anything. It will lead to contamination once again. And the other thing, because we use them in big poly houses very often, but the growth chamber is the best. But if you are doing them in poly houses, again, you have to be very careful that the insects and mites are not carrying propagules from one pot to another. And similarly for watering, you have to be very soft so that it doesn't splitter your soil from one pot to another. And in Australia, in UWA, when I was raising cultures, I was used to add measured amount of nutrient and water, and that too very gently in the cultures. And then we keep them on a bench rather than soil very often. Why? Because the soil might have these mites or other things which could bring infection in your pots. And in Australia we used to use the pots without a hole at the bottom, again because of the same reason that from the bottom no contaminant should come and join. And I said again, it's preferred to use a growth chamber than an open greenhouse because to prevent contamination, and the pots have to be kept at a distance because then the soil from one pot will reach to another pot, and the staff should be very well trained who's taking care of these pots. And it could be these bags. I often use these polythenes very often. Why? Because it could be a way, but depending upon the weather, you have different options available. Otherwise, sometimes it will lead to heating up of the pot, and then you don't get the multiplication if it is heated And so you have to decide according to your, sometimes you can think of the options of keeping them in the bag also. So a lot of people, when we have reached to this monophyletic classification, a lot of people are using this root organ culture also. I have not tried this, but this has really been helpful in reaching to this phylogenetic classification of Am fungi. In the beginning, the spores which were produced in this were of much better quality for rDNA amplification, but and this also gave the assurance that the DNA which we are getting amplified is not non-AM fungal DNA, but it still has disadvantages because sometimes in these cultures we produce much smaller spores in size, and it has not been possible. It doesn't work for many species. So again, like according to me, if you're doing the taxonomy work, I find raising pot cultures and especially the single spore cultures where the success rate is much less, but that gives you the best possible material, the material for starting your studies. So after you have got the single spore cultures for the species with which you want to work further, you must identify the spores. You must have, I said, 100, 200, 300 spores from that culture so that you can isolate DNA, you can make several slides, and you can observe the several structures associated. You can try germination studies and everything. So spore isolation, we follow the standard Jerdman and Nicholson. Sometimes for DNA work, we also tried the centrifugation method also. But and the same method we use, and we use series of sieves. We wash these spores thoroughly, and then it is backwashed into a Petri plate on a funnel. We filter these spores, and now everybody knows this procedure. And from the Petri dish, we carefully isolate the live spores. So this is an incomplete drawing from my side. I was short of time, but still what I'm trying to show you here is that you observe under the dissecting microscope. I'm just telling you the good practices which we follow when we study these spores, when we are planning to name any species is that what we'll do is from the Petri plates, we'll pick up all the spores and we make 3 watch glasses with a drop of water and at least 100 spores in each one of these 3, right? And then what we do is from the one, we make 5 slides. First slide is single PVLG drop, and we keep few spores from here on this, and we don't do anything, just put a cover slip. And sometimes, for taking pictures, we use this drop as it is. Then we pick a few spores and keep them here and put a cover slip, 1 PVLG. Then next slide we do what? Same way, pick up few spores, put PVLG, cover slip, tab this so that we can see the inside, what is happening. And the third slide, we take spores, we keep them in PVLG, and then we tab them badly, meaning little harsher, and we tab this thoroughly so that all the spores are broken and their walls are exposed, so that at least we can see what is there in the wall. Then we make 1 slide in Melzer reagent, and then we make another slide in Melzer reagent, but again tapped, so that we can watch the walls of these spores and the subtending hyphae. And this one more slide which I have kept here is that while sieving on the topsoil, we get the root fragments coming from the culture. So these root fragments we make clearing preparation for clearing preparation. This is how we do it. Then from the one watch glass coming from the same Petri plate, we make 6 type of slides. Then from the other watch glass, what we do is we again select the good spores, and we use these spores, at least 10 spores for DNA isolation. So what we do is we again select from these in an append-off and keep them in an append-off, and we use those for DNA isolation so that there is no scope for error of any type. And this third watch class, what we do with this is we take them back in a new sterile pot and try to multiply this again so that we have enough of specimen with which we are working for identification. So following this, we could identify, but by now we know that we cannot trust on morphological characters. They are unstable very often. And one species is known to produce several morphological types also. For the same species in the single spore culture also, you would see different structure. So we cannot trust them, but still, when we were initially doing it on the basis of morphological characters, we must study, and when we are reporting it in a paper, we are describing a species, we are expected to report all the morphological characters also. So let's begin with the phylogenetic analysis. How do we do that? And in this phylogenetic analysis, I took it first. Why? Because I said the morphological has taken a bit backseat, because the phylogenetic data is asked first when you are talking about identification of any Am fungi. So we usually use the DNA, the spores as a source of DNA. And again, this is again my picture with Manuela Kruger. I met her in Prague, and she is the one who designed these primers, which we follow, and they are called as Kruger primers. So I find this interesting when we are working with the scientists and when we personally know them, it gives me a fairer confidence. And when my students are stuck, I can directly direct them to that. You can ask her directly. So it gives a better feeling of being connected to the fraternity around when we're working with them. So this was it. So what did we do for isolation of DNA is that I said we take 10 clean selected spores in an append-off or in a tube, and with the help of a pipette or maybe a rod, you could crush these spores in the append-off, and we have a biored PCR in our lab. So you could add 100 microliter of biored buffer, mix it thoroughly, amplify using these primers. I said Kruger primers we used. We had both the end sequences, reverse primer, forward primer. We use these. And we did amplification following the standard protocol. And then for sequencing, we gave it for cloning and sequencing, we took help of Chromos Biotech Private Limited because we didn't have sequencer in our lab. And they gave us the sequence data, typical sequence data. And I said we had amplified the fragment. Which fragment? Small subunit fragment, ITS and LSU. So it had fragments from all these three. I tell you, it was not LSU sequences, it was not SSU sequences, it was not ITS sequences, it was all three fragments of those, because Manuela in her paper could identify many of this is the best work which has been done till now, and she could resolve many phylogenetic species on the basis of this primer. That is why we chose this primer. Though, otherwise ITS or maybe LSU could have been a better choice, because in INVAM, they use LSU sequences. So after having done this, we got sequences and bunch of sequences. And what we did is, first step was that in the notepad where our sequences were stored, we did Ncbi Search last, and we could identify many of these, and like few of these here I have highlighted with blue color, and we have also uploaded them on Ncbi so that we could get the accession number, because for publication they will always ask you for Ncbi accession number. So we took especially for those which we could identify, and there were several unidentified also. We uploaded all the sequences there. But some, like Glomus macrocarpum, Rhizophagus, intraradices, we could easily identify the sequences straight from the NCBI blast. But there were several unidentified sequences. We had to, identify them. And somehow we wanted to understand the phylogeny of these sequences. So we needed to make a phylogenetic tree. And otherwise, for curiosity's sake also, and we were in the process of building a database of the species which exist in our country, and we wanted to genetically identify them. So what we did is that especially for our unidentified species, we extracted all the sequences which refer to these Kruger primers, where these LSU, ITS, and SSU sequences were amplified. And for all the genera, so, at least I wanted to appreciate that my sequence fall there in the phylogen, right? So we isolated many sequences. They were about, I think, 327 or what. And this is this table from my paper. It tells you the NCBI accession number of these sequences, which I used for making a phylogenetic tree. I made two types of phylogenetic trees. One was with few sequences, where I published a paper. Another one where I built a huge phylogenetic tree. There I used a lot of sequences. And still, I could not identify some of the species. So how do we do for building the phylogenetic tree? I said we had sequences and we picked up some of the corresponding sequences from Ncbi database. And I used MAFT, and especially I used the MAFT server at EBI. And it is quite simple to use, well, I could not, because again, I said I left my stuff in India because I store my data on a hard disk, and I'm not carrying that hard disk. Otherwise, I would have shown you here my notepad also, where all the sequences are stored. But for building a phylogenetic tree, first we have to build make a multiple sequence alignment using any software. You could do it with Crystal, you could do it with MAFT, but I did it with MAFT because it can handle many more sequences. We had huge number of sequences, and I did it on EBI, this server. And it is quite simple, like any Google, I uploaded my sequences here, copied and pasted, or uploaded the notepad, and then I made a run. And I had few selections, which I cannot tell you here because again, I'm not carrying my notepad here, but I have the result with me. So this I used for multiple sequence alignment, and for making the phylogenetic tree, I used RAXML because I tried to follow the same procedure which Manuela had followed, whose primers which we are using. So I followed the same procedure. And since she used MAFT, I said I will also use MAFT, though it had certain additional features rather than Cluster, I told you already. And the software we used for making this phylogenetic tree was RAXML server, which is maximum likelihood phylogenetic analysis method. And in this, we did 1,000 bootstraps and GTRAMA model we used for bootstrapping and tree inference. See, what I'm trying to tell you with this is that the terms may be looking very complicated to those who have not worked with these, but it is not that complicated as it is seeming. So MAFT I have shown you, but I don't have a notepad. I could have run it for you, but I have the result. After MAFT, what happens? This result I got, right? So this showed me the complete sequences, and it showed me the conserved region, the one colored, which are same all through all these sequences that refer to the conserved region, and the variable region is lightly shaded because I had to take for snapshot, I had to reduce the size. So this multiple sequence alignment of 237 sequences I used for building a phylogenetic tree, right? So multiple sequence alignment is done. After that, we build a phylogenetic tree. And for building this phylogenetic tree, I have told you already, we used RAXML software, and that at Cypress portal. So this is the Cypress portal, which we use. Again, my data from here is gone, but I could introduce you with this. Here I have stored this as a, how to go about this is, once you have your notepad. So my screen is seen, and I just saved it here. Can you see it now? This website I want to show you. Yes? Yeah? My screen is visible to
Yes,
everyone.
Yeah, yeah,
It's
ma'am.
visible.
So I use this for building a tree. It's a online server which is available, which is I found is quite easy. And again, I said I use this because Manuela used this because I didn't want any variations in the methods used. So you could maybe log in. You can create a login for yourself, and I can show you this from my PPT also, right? And how did I do that? As maybe you can see, you have a login here. I have created a login. You can log in, and then you can have your data. I have a data file. I said Notepad, and this is the data file. I selected the data file. And I just want to make you familiar. I said this is a workshop, so at least you should get a hands-on on what is happening. So this was the data which I selected. And then what did I do is I then decided the task. And my task was to make a phylogenetic tree with this. And then I selected the input data again. It is asking me to select. And then after this, I selected the tool, and the tool it's available on this server, was again this RAXML. At least when you are familiar, if you try once on your own, I think you'll reach somewhere. And then I set the 50 parameters in this, and I told you already is here in the parameters, like you could bootstrap, you could add one. This is the model. I said I selected, I chose this model, and then I configured. I said 1000 bootstrapping. So why did I show you this here? Though at least I wanted to make you familiar is that it's not that difficult. Once you get the sequences, despite you don't have a sequencer and other facilities, you can do it online. Servers are available, and these parameters may be sounding very difficult, but not that difficult. Once you are determined, you can use their servers, they can perform this task for you, right? So after having made a phylogenetic tree, at this Cypress, I observed my tree. Again, another online software, which I like this software very much. This is iToll. It is a free again. You have to have a login here, and then you can create trees for yourself, and that create beautiful trees. I can show you a few of my trees because by chance here my trees are there. So this is the iToll website where my login exists. And I can show you my trees from here which are existing, still lying there. I missed my notepad. I couldn't locate that. So these are a few of the trees which I had drawn, like the one you can see. And you can do lots of things with this. The tree, you can remove whichever branch you feel that it is giving you confusion or maybe inaccurate. And to me, the most interesting thing which I found is my sequences, like I said, Glomus macrocarpum, they fell at the correct position in the tree. And I had built a small tree also for another paper that also you could do with this one. So it is a very simple, and you can modify it from here and make it as beautiful as you want. So this tree I have built with this website, and I colored it, and I could understand and study the phylogeny of this. Again, this tree I showed you already. This tree, again, I have built it from there. And few of my species still lie unidentified, but I could, somehow say that they are closer to glomus maybe, or we need to work further on them to identify. This work of mine, this database of 237 species, I have already published. It exists there, and the pictures I told you how I have drawn with this picture. So before I move on to the morphological methods for identification of Am fungi, I like to show you this, which I built on the basis of this work which we had done. This is a website which is getting older. Earlier this was on university web server, and now things are getting difficult there online. Everything is online, and those people are finding it difficult to handle. I have shifted it to some other server, and in this shifting process, it has lost few of the things, but still it works good. Like I can show you for many species here. If you search it by G, it opened all the species. 148, I said, you can open up details of any species, like I choose Glomus macrocarpum. You could open that and it will tell you that there are several popular names existing for this, but you should use Glomus macrocarpum, and this exists in these many areas. You can open the sequences of this. Maybe this is our sequence. And here, it is not coming now, but I had drawn a Google map, and here it will tell you the exact location from where in India it has been isolated. The phylogenetic tree, where you can select any sequence where you are interested, and it will take you to Ncbi for that sequence. So this is the work which we did at our UGC project and in our innovation project, and we reached here. But still, some of our species lie unidentified. And I told you already, this phylogeny becomes the basis for publication or doing any other thing, right? So that was about the phylogeny, and now I reach to the morphological analysis, and we mostly write the spore characters, and in the spore characters, we study spore color, spore texture, size, shape, subtending hypha, contents in the spore layers, smells. I told you all this already. That why germination is necessary, because with the germination we will determine whether this is scutellospora or a holospora. And we can, especially, I have this color card lying with me. Even now I used to do them manually, and I used to sit with the microscope and match the color manually. So, but we have to prepare all the data, right? The size, their arrangement, and the shape. So we have an access database for this one, and we recorded this for all the species, whichever we had. One interesting observation came out of this study. Again, I published this paper, and this was related to urbanization. The result was linked to urbanization. But what we had done here is that we were measuring the size, I said, again, for taxonomic purposes. And what happened is, what I observed here is, while making this heat map, is that spore size is also a very important characteristics may not be very useful for taxonomy, but when I'm studying the function of Am fungi in any ecosystem or in any agro ecosystem, what is happening is, like in my study, I had 3 regions. This was polluted, this was whatever. So but in one segment, if you see according to the spore size, this is 0, and this is increasing spore size. The larger species were disappearing altogether. So this interesting observation came from the same data, is that the size can be a very important character, at least in the ecological studies. But in taxonomy, very often, it helps you in making a preliminary observation, but for reaching to species level, it won't help you much, because for species, we need to know the wall structure, subtending hypha structure, germination layer. Those are the more important structures. So how do we identify? Once we have made the table of all these characters, I said we made the access database, then how do we go to identification? I tell you. So before, again, we had clearing preparations also. I thought I will tell you, but I think we are running short of time, so I have to pack up quickly. Do we still have some time? I'll take 5 more minutes and finish this. So these are the clearing preparations. What do we observe in clearing preparations in case of glomus is it produces typical H in the branching, and there's no clustering when it germinates. You get spores, and I already told you. So I think I can skip these slides because you know this already. So coming to identification, how do we identify? Meaning, how did we did this in present day scenario? There's one very good book by Blazowski. He's from Poland, right? I met him at Lisbon. And this book, it's a monograph of Glomeromycota. I don't know, I see a lot of online resources, and I'll show you a few right now in next 5 minutes. But I really like this book because it has big pictures, clear pictures of all the existing at that time. In 2012, this book was published, but it gives you very good pictures and very easy to identify. So we follow this book very often. All the characters, and it makes a meaning key of the characters, like if you have this size, if it is in clusters, and it makes a key. It gives you a very good key, and with the help of that key, again, I have this, I'm not carrying this book, otherwise I have shown you that key also, and PDF is not available, but still I thought I can show you. So it gives you a very good key on the basis of which, once you have noted all your characters, you can have a guess at least which species it could be. The first guess is coming from your phylogeny, and the other, you can use this book. And one very interesting website, which I use very often, very personally, is this one. And you know what is happening is this, at this website, you have every detail of at least common species. It gives you the, I said you make 6 slides. So in this 6 slides, you can see all the details of many species, if not the new ones. So if you're studying taxonomy, I would suggest you to visit this website. This is not in WAM, but it covers many species in very good proportions, it shows you. So this website is quite a favorite of mine, again made by Blazewski. And then, yeah, it gives you the methods. Then another one, I was talking of Chris Walker, and I said we follow a species list. They have also built a website. This website, I especially find this useful. How, when you're describing a species, what happens is this has both taxonomy and species list. Species list, I show you species list. I think I have just made this also online. Did I store this? Just hang on 1 minute. No, I didn't. Maybe I can try. You can see my screen now. Can you see my screen?
Yes,
Yes,
ma'am.
ma'am.
Yes, ma'am.
Okay.
Yes, ma'am.
Just hang on 1 minute. No, KM. Yeah. So here, the most interesting thing, which if you're planning to study the taxonomy, is this. They have prepared a species list, and I very often go to this species list. What is most interesting here is that you can get an update how many species are described till now. Again, made by my teachers, Chris Walker. And what it does is that if you want to study any species in detail here, maybe it has a complete list of all the species. Say, for example, your interest lies in any, maybe which one should I pick up? This, maybe this Ncbi Quota, right? If you're interested in this species, it will quickly take you to the original paper where it was described. Then apart from this, it tells you all the names and synonyms which are existing. And it suggests you to use this name so that there's no confusion. So these two websites, I find them very good if you are trying to identify a few species. And I'm back to my PPT now. Right? So this is the same one. I had recorded my screen that time, but I could show you from that. So I read to the next one. Yes, I didn't do that. I wanted to show you InVam also, but I think I missed it. Same way on InVam. Yeah, just hang on 1 minute. No, I missed that. So same way you can use InVam also by Morton, dots of Morton, they also list. But I don't know, somehow for my species, I used to find them missing at InVam. I found this Blazewski very good, and again, this original papers from Am Phylogeny for giving them name, confirming their names after the phylogeny to be very useful. So I think I need to wind up, though I wanted to tell you about a few more of our things we are doing on the in the metagenomic data also, and from the rhizosphere we are trying to identify Am Fungi, but I got to skip this because I think we're short of time. I'm finished with my time. So I wanted to dedicate all my talk to my supervisors, Professor K.G. Mukherjee, where I began in 1988. Then Professor Lynette Abbott, with whom I worked in University of Western Australia, and Professor Chris Walker, who keeps enlightening me and giving me updates. Look, this is happening. This is what you people are doing in India. And he told me how you people are identifying. I tell you, they look at the sport and then they go to the website and match the picture and say, this is this species. So his talks usually are eye-opening to me. And I owe a lot to them because they have really trained me to what I am now. So I worked on several projects. Even now I'm working on one international project. And I just want to dedicate this to this funding, which I received from University of Delhi, UGC, and now presently I'm working on ICOMET. It's an international mycorrhizal collaboration of mycorrhizal ecological traits. And we have a scientist from 26 countries working on this. And my first paper from this project is coming with a lot of authors around. So my basic question again falls here. So I think I would like to, let's hang on 1 minute. So I need to stop sharing now, and I'm done with this till here. So that was it, which I could plan being away from home. Just 1 minute. Screen sharing is not stopped, isn't it? Yeah, it's gone. Screen sharing is gone. So we are face to face now, right?
Yeah, ma'am.
Yeah. So that was it. But I'm sorry that for a few of the things I had the handicap, but because I'm not at my home right now, and I could not arrange for a few things, but still I tried if I could inculcate the interest among the students or the fellow researchers who are there with us. I would be very happy to answer any questions if you have any.
Okay, thank you, ma'am, for an excellent talk. May I invite questions from the participants? Please, Shika, please introduce yourself. You can switch on your video, introduce yourself, and ask your question.
Yes, ma'am. Yes, ma'am. First of all, a very good evening to everyone. Good evening, ma'am. Ma'am, it was a very informative lecture. I'm Dr. Shikha Prasad from Deen Dayal Upadhyay College. I'm teaching in Deen Dayal Upadhyay College faculty, Department
Thank
of
you.
Physics,
Yes.
University of Delhi. Ma'am, first of all, it was very nice to see you, though in a virtual world, but live, because I've
Thank
been
you.
watching your videos on concepts of bioinformatics, and it's very informative and making my students also to see them. So
Thank you.
that's very
Thanks a lot.
nice. Those videos are very nice. Ma'am, basically, my PhD was from forest ecology. So
Okay.
I basically worked on native and non-native tree species to see the ecosystem processes, difference in ecosystem processes under native and non-native species. So ma'am, my question is very general, though I didn't work on Am fungi, but it was very interesting to me. And I was curious to know, ma'am, does native and invasive plant species, they differ in their interaction with Am fungi, or ma'am vice versa, like does Am fungi differ in interaction with native and non-native plant species?
Yes, because of course in any ecosystem, some of the species are dominant ones. They will know, and the same happens even among Am fungi. Some of the species they would occupy all the area very quickly. Though it is tried and it has been studied, especially with reference to invader plants. The invader plants, they
Yes,
grab the ecosystem with the help of Am fungi because they
oh,
colonize, they become more fit in the new ecosystem. So the invader plants, especially it has been studied with invader plants, is that most of the invader plants, they are mycorrhizal.
okay.
But some of the species, of course, among the Am fungi also, some are very fast growing and invading. They would quickly occupy all the area. And same way, I just touched upon this,
Yes.
is that we should not add inocula unknowingly because it is going to disturb the native species, and it may be an invader sort of that it doesn't allow the other, because you don't get maximum benefit unless you are in your natural situation. Right? I hope that was what you were asking.
Yes, ma'am. Yes, ma'am. Actually, you mentioned now that Am colonization should be done on native flora. So that's why
Yeah,
I got curious to know about that.
yeah. meaning it is happening, you will see
Yes,
several
ma'am.
papers. They emphasize that there was one paper by, again, my UWA colleague. She
Okay.
said that farmers should not be recommended to apply this AMF as biofertilizers because it does not give you any result. And the main reason was this, because unless the native flora is studied and the context dependency is checked, it is not going to give you any result.
Okay.
So this is quite important to know about the native flora as well when we are applying this as well.
Okay. Thank you so much, ma'am. Thank you so much.
My pleasure.
Next, we have Asha West. Asha Beast. You may please ask your question. She has raised her hand. Okay, then we have Nonia. Is it Amanso? Is it you? You want to
Yes,
ask a
ma'am.
question? Yeah.
Yes,
Please
yes,
put
ma'am.
your-
It's me. Good evening, ma'am. It was such a
Hello,
nice
Ravi.
presentation. Thank
Yeah.
you. Ma'am, as you said that for identification of AMF, we need to have live samples of mycorrhizal spores. But how do we identify that which AMF spore is live and which one is dead?
Yeah. Yeah. So, that's the primary step. But if you have observed any ring in the Petri plate after spore isolation by Judman and Nicholson, the live spores will always be transparent. See, I tell you, do I draw it for you? Just give me 1 second. Well, so maybe I can draw it also, but if you observe them, the dead spores, first of all, they will not be in proper shape, right? And the other thing is the dead spores, you will always see dot-like structures on them. That refers to infection, bacteria or anything in the feed spores. And third thing is the cytoplasm is withdrawing, right? So if you straightforward want to pick up the live good spores is they will always be transparent and their cytoplasm will be uniform, right? And in field samples, most of the spores, they are dead. This is what I told you. This is the one error which I observed is happening very routinely. People try to identify from field spores, but the field spores are usually dead because they're dying, they're under the process of recycling. So, meaning what you collect basically, otherwise close to the roots, some maybe. So when you want to pick up, you have to look for clean, surface clean, and the surface clean you get when you do thorough washing, right? And then you have to pick the spores which are transparent on whose cytoplasm is clean. This is what you were asking?
Yes, ma'am.
Yeah.
And suppose if we are trying to find the spore density in a soil, should we consider both live spore and dead spore, or just live spores?
That's up to you. What is the aim of your project? It is good to. I tell you one synonym here. When I was preparing this database, because Chris knew that what is happening in here, he said, why don't you add a method on this identification, whether a morphological method is used or a phylogenetic method is used. The moment I added that criteria, I could know that most of the identification has been done on the basis of morphological characters in India. So that is not acceptable to anyone. So maybe it is liable to wrong identification. So your question was, what did you say? Yes?
I asked that if I'm trying to find spore density in a soil, should I consider both live spores and dead spore, or just live spores?
Again, I tell you what. It all depends on the aim of your project. If you are studying recycling, the dead spores also has a biomass, right? But if you are talking, it all depends on the aim. But still, you will be able to distinguish. When you are picking up spores carefully, you will be able to distinguish. And live dead, nobody will agree to your paper if you are sending this for publication. Live dead, you would have to conclude on the basis of germination test, or there is one FDA test. Right? You have to show them, by your observation, you cannot reach to any ecological conclusion. You have to perform proper germination test when your result wants to talk about the live and dead, meaning you were talking about basically the effective biomass, right?
Thank you, ma'am.
Thank
Okay.
you.
Next, we have question from Asaya, AJK Asaya.
Asaya, yeah.
Good evening, madam. Good evening, madam.
Yeah. Hello. Yes.
From Tropical Forest Research Institute. I worked with
Sorry,
Dr.
I can't hear you. Can you be a bit louder?
Madam, this is AJK Asaya from Tropical Forest Research Institute, Jabalpur.
Jabalpur. Okay.
Yeah. Madam, I want to ask, in problematic soil, it is recommended to use Am Fungi. So, which one we have to use? Since in ravines or in coal mine overburden dumps, whenever we are going to do plantations, it is recommended to use some Am Fungi. So, how to isolate the native ones?
See, what is happening is that your voice interrupted in between, and I couldn't get your question. So you would have to repeat and be a bit louder, please.
Madam, I am posting it. Okay.
Yeah, that would be better. Maybe you can write side by side.
Yeah.
So it is coming in the text box. Yes.
Meanwhile, ma'am, there's another question from Hageya Kang. It asks, how does season affect the diversity or sporulation of Am fungi? You can see in the
Wow.
chat box also.
So she has written it in the chat box.
Yeah.
How does season affect the diversity or sporulation of Am fungi? The first answer, or the very simple, straightforward answer to this, is that the season affects the growth of the plant with which it is growing, right? So that is one thing. And apart from this, temperature also has been shown to have some influence, the environmental conditions on the sporulation, moisture, temperature. So the season is going to affect, but season more depends on the host species, whether that season is suitable to that host species or not. Okay. Who was this? Thank you for your question. I hope I answered you.
Yes, there's another question coming in the chat box itself.
Yeah. Yeah, I will read this, yes. Again, I would say for this one, Dr. Basaya, sorry, I'm pronouncing Asaya, Dr. Asaya, I hope I'm pronouncing you right, is all depends, meaning I would straight say, first analyze the native species of Am fungi existing there, right? First isolate the native flora, and you would know which species is flourishing best there, and raise a culture of that, and I would suggest using that in that area, because this is how we decide. I don't recommend using inoculum from outside. If you still want to ask, maybe you can write again or try speaking.
You have another question, ma'am?
Yeah, maybe first let's attend. He's a very senior scientist, Dr. Asaya. You still want to add something, Dr. Asaya?
Madam, thank you.
You would have to write it. It's not coming. So Sunil Kumar is asking, Am fungi can be used with bacteria? Yes. Yes, why not? It has been practiced. They have been used as consortia. Yes. They are, I think they are most successful in being used as consortia. So very sorry for Dr. Asaya, his power is gone. So, but I maybe you could connect to me later on and we'll have a good discussion about this. I will request this Alvin to share a video of this with all the participants so that, and with me as well. She is recording this.
Yeah, okay, ma'am.
Some are asking for the PPTs, and I think rather than PPTs, video is more useful. You get a voice also.
Ma'am, you have another question from Asa Vish.
From, yeah,
Yes, regarding the biodiversity, apart from
Asha.
yeah,
So let me first take the privilege of a very good morning. As we always, as your is immensely enlightening. My question is regarding the biodiversity. Apart from morphological and phylogenetic identity, can temporal spectrum also play out and show polymorphism? Since they show. See, all the factors, if you're talking of morphology, not only temporal, all the features they affect and they are not reliable. Sometimes the culture conditions change the morphology. Their morphology is not stable. That is why, on the basis of several discussions, it has been decided that phylogenetic characters were to be first considered and they are most trustworthy. And you have to have several slides and type specimens to confirm their identification. Yes, temporal relation also has been shown. Now host specificity also is shown for several species. So the new information is being added. Temporal variation also is there. Ajay Sani, can 10 spores infect? Okay, just hang on 1 minute. As Ajay Sani is asking, can 10 spores infect make 12 IP? Is it possible? What does that 1200 IP means? Ajay.
he means to say it infective propagules. Can 12 spores infect? Ajay Sani, can you make your question clear? If your network is clear, you can come online or switch on a video and ask.
Yeah, Evelyn.
Yes, sir.
Yeah, actually, this question is coming from the last presentation by
Yes.
Mahabir Sharma.
Yes, sir.
In his presentation, he showed that if you have 10 spore per gram soil, you can have IP amounting to 1200. So,
Yes,
and
sir.
he explained also that how the IP will be 1200 or 12 million per square meter, per cubic meter. So better the participants should revert back to Mahabir Sharma because he defended and he answered his findings. Okay.
Yes,
Yeah.
sir.
So
Yes,
that
sir.
should not be, that question should not be put here. But from my side, I can add that infective propagule includes the mycelia present in the soil, the mycelia present in the root, as well as spores. So it is possible to have lesser number of spores, but more IP. Thank you.
Thank you, sir. Ma'am, we have one more question from Aparna Notial.
Yeah.
Which part of the roots should be taken for best results for seeing AMF as arbuscules and vesicles under the microscope, and what plants specifically?
So part I would say that you could leave the tip portion in the root, especially they take the fine roots and leave the tip portion of the root, and you then cut it into small fragments and use those roots. They will give you good result. And you can use trigonella roots, or if you want to use other roots, you can use any root, because you have to go for this clearing of the roots. That will remove all the pigments from there and you'll be able to observe. So, but if you want to use this in the laboratory for classroom teaching, maybe trigonella roots, they show you good result. So
There was
Deepika
a
Dalal
question.
has asked a question.
Yes, she asked a question, but you have already explained that in your presentation. She says, for identification of native AMF species, you said the spores are mostly dead from the field samples. Is it that rhizospheric soil also has dead spores?
Okay,
Does
where
the
is the
rhizosphere?
question?
She posted at 552.
552, okay. Yeah, Deepika Dalal. Native, you said the spores are mostly dead from the feast. Yeah, The rhizos, see, it cannot be always that the dead spores are present. I was talking from identification point of view, right? The spores we isolate, live spores also, young spores also, all come. But what I have observed is that mostly because the dead spores, they are larger in size, they are commoner in appearance, people pick those spores. It cannot be that the live spores are not present. It's a continuous process. These spores are being produced in the rhizosphere. So you will have all type of spores. It's not that only dead spores will be present, right? In the good rhizosphere, you will have all type of spores. Okay, Deepika?
Dr. Meghna, there's a question from Dr. Meghna, ma'am.
Yeah, where is it? Where is that?
Hello, madam. Can you
There is
hear
one
me?
from P.
Am I audible?
Non-King. She says, how can we and vesicles? So you see wall layers in the spores and not in the vesicles. If you are talking of the spores, intraradical spores and vesicles, you see wall layers in these spores, right? Then Nonya, is it okay to consider only morphology for identification without considering for genus level if the spores are healthy? Yes, to the genus level you can say, but you cannot go to the species level without phylogeny. Yes, up to genus you can do it.
I think we had a volley of questions even for you. It was an exhaustive one question session.
Thank you that people found it interesting and that could kindle the query in their mind, and that was my main purpose of giving this talk.
Thank you,
And I
ma'am.
am available to everyone. You can talk anytime, or if anyone wants to collaborate in any project, most welcome.
Okay. Singh sir, you want to add on anything?
No, I gladly accept the offer for collaboration. Evelyn?
My pleasure.
Yeah.
Yes, sir.
Yeah, Evelyn and Oi, both of you, take advantage of this offer.
Yeah, sure.
Yes, sir. Yes, sir.
Yes, sir.
Acha, thank you, ma'am. But before I ask Dr. Uedai Nimaso to propose vote of thanks, let me make a small announcement to the participants. Actually, this workshop was organized for 3 days, and we were supposed to have Professor Bernard Rodrigues from Goa University to deliver his talk tomorrow. Unfortunately, he got COVID and he was in ICU just 2 days before. So he had been advised bed rest, total rest for 10 more days. So we are very eager to listen to him, and I'm sure all the participants would also like to listen to him. So we are not canceling his talk. We are waiting for him to recover, and we will again inform you all through your registered email. Probably by mid of February, we will again ask him to deliver his talk. So tomorrow we won't have any lecture, and we will inform later on. Thank you. So now may I invite Dr. Uedai Nimaso, Assistant Professor in our department, to propose vote of thanks. Ma'am, please.
So do I leave now? Thank you everyone for inviting me here. And
no, ma'am, there will be a formal vote of thanks.
Okay. Please.
Just couple of minutes.
No problems.
Good morning to ma'am, and good evening to all of you over here in India. Most respected head of the department, Professor R.K. Singh sir, and our esteemed speakers of today's technical sessions, Professor Manju M. Gupta from Department of Botany, Sri Aurobindo College, University of Delhi, faculty members of department, as well as faculties or members from different college and university, and my dear students. participant. I'm Dr. Oiday Nimaso, Assistant Professor, Department of Botany. It is my privilege to extend a vote of thanks on behalf of the organizing committee. First of all, I would like to extend my hearty congratulations and thanks to today's STV speaker, Ma'am Professor Manju, for her excellent talk and presentations. Thank you, ma'am, for taking out your time, especially joining us from abroad and enlightening us about different methods of arbuscular mycorrhizal fungi identifications. And it was really, really interesting to hear from you. And it was really insightful interactions. Thank you, ma'am. Thank you so much. I would also like to extend deep gratitude to all the faculty members, research scholars, and students, participants from different colleges and universities for attending the technical sessions and encourage and present the technical sessions actively, very actively, as I believe without your presence and interactions, the session would have been just a PowerPoint presentation. Thank you all for making it useful. I'm sure we all will get the benefited from today's talk, from the excellent talks given by Ma'am Manju. And I would also like to thank the strongest pillars of the department, beginning with Professor R.K. Singh sir, head of the department, as well as chairman of the ongoing project, and Professor Abhaya Prasad Dast. Most of them are not joining today, but I would like to extend my thanks to them. So I'll be mentioning them. In continuing the Professor Abhaya Prasad Das, then Professor Sumpam Thangjang, Dean Faculty of Life Sciences, then Professor Huitak Sir, Dr. I am Victor Singh. Sir, thank you all for always being there to support, to guide, and especially making this workshop a very successful one. And also, lastly, I would like to thank the building blocks of the department, beginning with Dr. Evelyn Haikam, convener of ongoing workshop, then Dr. Tenya Rina, then Dr. Tonglong Wanpan, and Dr. Vibhuti Dast, and also all the staffs of the department for making today's technical sessions a grand success. So thank you all of you from the Department of Botany, Rajiv Gandhi University. Thank you.
Thank you. Thank you, everyone. Thank you, Dr. Rajiv and Evelyn and everyone for being here and making it a success. Thank you.
Thank you, ma'am. Thank you.
Yeah. Do I leave now?
Sure,
Thank you.
ma'am.
It was so nice to see you. Yes. Bye. My pleasure.
Bye, ma'am. Bye.